So welcome back, friends. So we talked about the mechanism of action and antibacterial spectrum of the macrolides. Now this is the part B of the macrolides, and in this video we are going to talk about the resistance which are activated against macrolides. So let me write resistance. Macrolide resistance. Macrolides resistance or resistance against macrolides. Now there are many ways of the resistance which are activated against macrolides. So whenever the bacteria process or growth of the bacteria process are stopped, so bacteria release a kind of chemicals. These chemicals are sent to the suspected to some other bacteria which are present in the environment, and these bacteria then produce and these bacteria we can say then resist. Or what actually happen, let's suppose this is a bacterial cell. There are many ways of the resistance. So the first resistance, first of all, let me talk about this is the first one resistance. The first one resistance is mutation, or we can say alteration, or we can also say modification as well, or modification. Now what actually happened in the first case? In the first case, let's suppose this is a bacterial cell. Inside a bacterial cell there is a genome. So these are the genome of the bacteria. And there is also a plasmid as well. And all of you better know that plasmid is a self-replicating DNA. It is obviously in a circular form. This plasmid will be replicated in this form, means they will be replicated in a rotatory form. So when it is replicated, so then it produces a genes, we call that arm genes. It produces a arm genes. This arm genes will be produced and shift to the genome of this bacterium. So this arm genes, so this is the actually arm genes. So they produce the arm genes and shift to the genome of the bacterium. So when they shift to the bacterial cell, bacterial genome, so what actually happened then during translation and transcription, an enzyme is produced. We usually call that methylase enzyme. Methylase enzyme, fine. So what actually do this enzyme? This enzyme produce a 50S ribosome, but in a mutated, or we can say they can produce mutated 50S ribosome. So in the previous video lecture, if you see the 50S ribosome will be in this form, but due to mutation or due to the presence of this methylase enzyme, this 50S ribosomes will be in a red form, or we can say this, let's suppose this red one represents that it is mutated, but the blue one is the same. We can say there is no mutation in the blue, so means there is no mutation at the red. This is the messenger RNA. What actually happened due to this mutation? The drugs, means the erythromycin. Let's suppose this is the erythromycin. The erythromycin will lose their affinity and they not bind to this drugs because they are mutated. What? They are mutated. So when these drugs are mutated due to the methylase enzyme, so these macrolides lose their affinity or lose their ability to bind to these drugs. So when they are not binding, so then translocation will be occur again. So the translocation process will be occur and again the protein will be synthesized. This is the first way of the resistance. Now let's talk about the second way of the resistance. What actually happened in second way of the resistance? In second way of the resistance, there is efflux pumps production. Efflux pump production. So what actually happen in efflux pump production? I already explained it in a very high means I already explained it. So I already explained the efflux pump. So let me again explain it a little bit. So what actually happened in the efflux pumps? There is a genes which we call that MIF-A genes. MIF-A genes. So let's suppose this is a bacterial cell again. Then these are the genome of the bacterium. Fine. And here or somewhere of the genome there is a, let's suppose this one represents the MIF-A gene. So due to the presence of this MIF-A gene, this MIF-A genes produce these efflux pumps. So how these efflux pumps will be look like? These efflux pumps will be look like in this form. So due to the production of these efflux pumps, we also call that a transmembrane protein. We also call that a transmembrane protein. So we also call it transmembrane protein, or we call it channel protein as well. Channel protein, or we can also call that efflux pumps as well. So what actually happened in this case? In this case, let's suppose this red one represents the drugs, or we can say these represent the erythromycin or clarithromycin, or let's suppose we can say these are the macrolides. So these are the macrolides which are internalized inside the cell through a porine channel or through a passive diffusion. So when the drugs are internalized inside a bacterial cell, so due to the production of these efflux pumps by MIF-A genes, what actually happen, they kick out or pump out these drugs through these channels. So through these channels, these drugs will be pumped out. So these are the macrolides which are pumped out. Fine. So due to the production of efflux pumps, they will kick out the macrolides which are present inside the cell or which are internalized inside a bacterial cell during passive diffusion or through a porine channel. So that's all about the resistance which are activated against the macrolides. So now let me write the side effect of these drugs. So that's all about the resistance. Now let me write the side effect of these drugs. So if we explain the side effect of these drugs, so whenever we are giving these drugs for a longer period of time, so there will be a side effect of these drugs as well. So which kind of side effect? Let me write here. Side effect. So which kind of side effects are produced whenever we are giving these drugs? The first side effect is GI disturbance, gastrointestinal disturbance. This is the first one. The second one is jaundice. The third is ototoxicity. And there are some other side effects, but these are the common one. Fine. So that's all about the macrolides, side effect, mechanism of action, resistance, antibacterial spectrum, and some other discussion about the macrolides as well. So I hope I explained it in a very huge amount, but still if you have some questions arise in your mind, or if still there is some missing points, so write it into a comment. I will try my best to give you answer or solve your problem or clear your problem as well. So please like the video, share the video, and whenever you want more videos lecture, or whenever you want more antibiotic videos lecture or some other video lecture, so please subscribe the channel. Then you will get the updates of the other videos which are related with the antibiotic drugs. Fine. So thank you so much.